So, good day everyone. Hello. So, I'm gonna report about the secret life of tube worms, a journey through annelid reproduction. Marine invertebrates called tube worms are classified as Polychaeta, which is Greek for many hairs. So, these organisms can be found in a variety of marine habitats, including shallow seas, cold seeps, and hydrothermal vents. The peculiar life cycle and reproduction of tube worms are some of their most intriguing features. So I will go into great detail about a tube worm's life cycle and reproduction in this presentation. For the objectives, first, so to understand the several stages of a tube worm's life cycle, such as larval growth, settling, and adult form metamorphosis. Second, to explain the creation of gametes, fertilization, and embryo development as they occur during sexual reproduction in tube worms. Lastly, to examine how tube worms have adapted to survive and reproduce in their particular settings, such as cold seeps or hydrothermal vents. So, there are so many species of tube worm, but here we will focus on the species called Hydroides elegans. So it is a type of marine polychaete, which is also referred to as the elegant feather duster worm or the calcareous tube worm. As you can see in the figure or the photo, it looks like a feather duster. Somehow looks like a feather duster in an elegant way. And throughout the world, coastal and estuarian habitats are home to this member of the Serpulidae family. In order to cling to hard surfaces like rocks, shells, or man-made structures, Hydroides elegans builds a characteristic calcium carbonate tube-like structure. So this tube protects the worm and acts as its residence. The worm itself has a segmented body that protrudes out of the tube in the form of radioles, which are feathery appendages or attachments used for filter feeding. So in the next slides, I will show the morphology of this species so that you will appreciate more its parts and what are their functions also. So external fertilization takes place and the embryos or larvae quickly grow in a straightforward dish of seawater. So I based this presentation in a journal, but we will see if external fertilization really takes place during their natural reproduction in their natural habitat, which are in cold seeps or hydrothermal vents. But in the journal, they use external fertilization, the spawning, which is an analytical process in which they release thousands of eggs and sperm simultaneously. The reference journal partially described morphological and molecular characteristics, the cleavage stages, and early embryogenesis, as well as creation of the feeding larva stages, segmentation, and emphasis on metamorphosis and juvenile life stages. So I'm gonna present here a thorough staging of the life cycle of hydroids from fertilization to sexual development, as well as insights on how these model might be used to improve our comprehension of a variety of important and fundamental biological issues. So the hydroids morphology and life cycle. So the figure A is an adult. She is sexually mature. And then, as you can see in the figure A, so the symbol AB, it is the abdomen. The BC is the branchial crown. The letter C part is the cola. OP part is the operculum. And then under the operculum or symbol PD, it is the peduncle. And then PG, the like a tail something, it is the pygidium. And then we have the radial under the bronchial crown, symbol letter R. And then TH, the thorax. So the abdomen of Hydroides elegans is responsible for secreting the calcareous tube in which the worm resides. It also functions for protection and anchorage. Tube ripe and growth also functions for respiration. While the primary site for respiration in Hydroides elegans is the gills, located in the thoracic region, the exchange of gases between the worm and the surrounding water can also occur through the thin body wall of the abdomen. The bronchial crown of the tube worm, located at the anterior end of the body, is primarily responsible for respiration. It consists of feathery appendages called radioles that extend out of the tube and function as gills. So these radioles facilitate the exchange of oxygen and carbon dioxide between the worm and the surrounding water, allowing for respiration. The operculum functions for tube closure, also for defense against predators, maybe. Also functions for water current regulation. The peduncle functions for attachment and anchorage of the worm towards hard surfaces, flexibility, and also for movement. The pygidium functions for waste elimination, so it somehow is the anus part of the tube worm. It also functions for tube closure, protection, and tube anchorages also. The thorax functions for locomotion. It houses the internal organs such as the digestive system, circulatory system, and nervous system. Also for reproduction, it houses also the reproductive organs. And for sensory perception, the cola is for feeding, so it somehow is the mouth of the worm for defense and sensory perception. And the figure P, it shows an illustration of the main life history phases of Hydroides elegans by phasic life cycle. Interaction with single or multi-species biofilms with bacteria causes metamorphosis of the larvae. So from the fertilization, we have now the zygote, gametes and zygote of Hydroides elegans. When hydroids eggs are fertilized, there are no visible morphological changes, unlike the gametes of other marine invertebrates such as sea urchins. So the figure A shows the sperm of the Hydroides elegans, figure B shows the unfertilized egg, and figure C is the zygote or the already fertilized egg. The duration of each stage can vary based on factors such as temperature, food availability, and environmental conditions. While larval stages may last for several days or weeks, the settled worms can live for months or even years, continuously growing and reproducing within their tubes. It's important to note that more specific information on the precise duration of each stage in the life cycle of Hydroides elegans may require further research and specific studies dedicated to this species. Now, from the zygote, we have the successful fertilization, we have the embryonic cleavage stages in Hydroides elegans. So, early Hydroides cleavages are equal sized, as you can see in the figure. Unlike other Spiralia members and even other annelids, at the four-cell stage, the D quadrant of the embryo lacks morphological differentiation from the other quadrants. As you can see, no same language. It's just the same. No morphological changes and differentiation from other quadrants of the cleavage. The first division during which the spiral pattern is visible is the third cleavage, which results in an 8-cell embryo, as you can see in the figure C. The division is a left-handed sinistral twist. The daughter cells are born on the opposite side of the macromere as a result of the cleavage spindle rotating, providing the impression that the animal pole of the embryo's blastomeres have turned counterclockwise in relation to the vegetal blastomeres. The 8 embryonic cells finish the subsequent cleavage cycle simultaneously, giving rise to the 16 cell stage, as you can see in the figure D. In contrast, though, the blastomere divisions leading to the transitions from the 16 to 32 cell stage, which can you see in figure E, and the 32 to 64 cell stage in figure F, do not happen simultaneously. Now, the vegetal pole of the embryo is shown pointing toward the page, while the animal pole is shown pointing outward in each panel. So, from cleavage, we have now the gastrulation and larval stages in Hydroides elegans. So, following the full cleavages of the 64 cell stage embryo, now known as a blastula, the creation of the gut via invagination at the vegetal pole commences. So, the embryo also develop cilia, the white arrows on the figure B to C, B and C, and hatch to start their free swimming stages. They then generate an apical tuft that is typical of many marine larvae. The yellow arrow on figure C, that part is the apical tuft, as well as a thick ciliary band that will later form the prototroch. The entire gut is tripartite or consists of 3 parts with a distinct foregut, middle gut, and hindgut. So the entire gut is tripartite with a distinct foregut, middle gut, and hindgut. The trochophore larva can then start eating at around or post-fertilization. The larva has now developed into a segmented ethicate. However, the body does not grow any more segments until after metamorphosis. So between 5 and 6 days after fertilization, the larva reaches competency, as you can see in the figure F, at which point it has 3 complete body segments and differs morphologically from the pre-competent larva. In a nutshell, competent larvae have bigger overall body sizes and a more cone-like body profile from the prototroch to the tail than do pre-competent larvae, which seem more rhomboid. So the head is more clearly separated from the body in competent larvae because there is no tapering or gradual sloping at the front areas of the body, unlike in pre-competent larvae. The head is more clearly separated from the body in competent larvae, as you can see in figure E. After that, the capable larva is prepared to land on a substrate and go through transformation. Gastrulation in Hydroides elegans larva phases. A blastula with cilia. The blastocell is outlined by a dashed white line, as you can see in figures. So figure B shows that the animal's oral side is pointing downward at mid-gastrula. The developing prototrochs ciliated cells are indicated by white arrows. Figure C shows the hatched glate gastrula. Developing prototroch ciliated cells are indicated by white arrows. The apical tuft cilia are indicated by the yellow arrow. Figure D shows a trochophore larva feeding seen from the side. Figure E shows a segmented netocate larva in an aerial perspective. Figure F was an oral view of a larva that is capable of metamorphosis. Now we have the Hydroides elegans undergoes morphological alterations that are related to bacteria-induced metamorphosis. So figure C, protothroxilia shed, eversion of the collar. Figure E, food groove cells are shed. Figure F, the first lobes are beginning to form, dashed white line. Figure G, lateral growth and development of the anterior lobes. Figure H, youngster after 24 hours of metamorphosis. Figure I, a juvenile at the 1-week post-metamorphic stage. Head inset and branchial crown elaboration. Contact with heterogeneous multi-species or monospecies biofilms causes metamorphosis. The larva makes brief, reversible contact with the settlement surface while investigating it, giving the impression that it is crawling along it while wriggling or flexing its body by contracting the muscles along the body wall before returning to a leisurely swimming speed.
Slowing swimming speed and probing of a surface are characteristics of settlement in response to a naturally occurring biofilm produced from seawater. If appropriate, the larva sticks to a surface, as you can see in figure A, and starts to secrete a primary proteinaceous tube, figure B dashed line. It also starts to lose the prototrochlesia, as you can see in figure C, marked with A asterisk, that are no longer necessary for swimming. So the larva sticks to the surface, starts to secrete a primary proteinaceous tube. Major tissues remodeling takes place as a mineralized secondary tube is deposited. The interior region of the larva's body changes shape to form a cola, figure 5D. Food groove cells between the prototroch and metatroch are shed. So, post-metamorphic juveniles, figures H to I, continue to develop and the anterior part of the body, developing the operculum and main radioles for feeding on the branchial crumb. As you can see in figure I, in 10 to 14 days. Within a week after the transformation, they reach a length of several millimeters while continuing to deposit their calcareous tubes. As you can see in figure 5I, reaches a length of several millimeters. So, summary of hydroids development from fertilization through sexual maturation. Individual stages are used to label the major development phases. The stages of metamorphosis, the figure M1 through figure M7 are the attachment to substrate, or the figure M1, primary tube formation in figure M2, prototrochlesia shedding in figure M3, collar eversion in figure M4, food groove shedding in figure M5, onset of lobe formation in figure M6, lateral extension and elaboration of anterior lobes in figure M7. So now we will go through the reproduction of Hydroides elegans. So a tube worm sexual life has been investigated in the deep waters, and now we will see if external fertilization or spawning is really the reproduction strategy of Hydroides elegans in their natural habitats. So the females of 5 species of deep sea tube worms fertilize their eggs internally with the aid of a hook-shaped sperm bank in their genital tract, according to research by Professor Paul Tyler, graduate student Ana Hilario, and colleagues from the University of Oregon. So it contradicts a process known as broadcast spawning that is used for external reproduction. The widely accepted theory that these tube worms reproduce by freely dispersing their eggs and sperm into the water, a process known as broadcast spawning, common in marine invertebrates like corals, is refuted by the researchers' findings, which were published in the February 2005 issue of the Biological Bulletin. According to their hypothesis, the sperm either swim into the spermatica, which is a hook-shaped sperm storage area where their female stores sperm and eventually fertilizes her eggs in vitro before she lays them. So they hypothesized that the sperm either swim into the spermatica or are gathered by the female through an unidentified method. Until the female tube worm releases them, fertilized eggs do not start to develop into embryos. The researchers discovered that fertilized eggs do not start to develop into embryos until the female tube worm releases them by studying tube worms' eggs after the females had spawned them. So, for summary of key points, trichophores are the planktonic larvae that Hydroides elegans used to start its life cycle. The trichophore larvae settle down and undergo metamorphosis via interaction with natural biofilm to become nectocate larvae, a benthic form. During the tube-dwelling period, the nectocates carry on growing and maturing into fully grown adults. Female tube worms can store sperm in the spermatheca, which is also where her eggs are finally fertilized, So before she spawns them and they develop into embryo. Following fertilization, the eggs grow into trochophore larvae which trigger Hydroides elegans life cycle once more.